Well, good morning, everybody, and welcome to Coffee Chats with Experts. Today's topic is cell sorting tips for success. And so I'm very excited to welcome our lovely panelists who are joining me this morning. Morning, everybody.
Morning, Laura. Morning, everyone.
All right.
Morning.
And so first up, I would like to introduce Chris. Hey, Chris.
Hey.
So Chris is based in California, same as me, originally from somewhere else though, right, Chris?
Yeah, a little bit, yeah.
The other side
of the pond.
Yeah. That's right. And so Chris has been working with Bio-Rad's flow and cell biology platforms as an R&D scientist and as a product manager on the business side. And he started at Bio-Rad about 5 years ago. So welcome, Chris.
Thank you. Glad to be here.
Yay. And next up we have Alan. Hey there, Alan.
Hi, how are you doing?
All good, all good. And another non-American or non-originally American person joining us this morning. So it's great to have you. I hear you're based down there in Southern California. Is that right?
That's correct, yes.
How's the weather down there today?
Same as always. Awesome.
Right? That's why we moved here. And so we're really excited to have Alan join us because he has been working as an application support person in the field there for over 7 years, supporting people in flow cytometry. And he's been with Bio-Rad for about 4 years now. So welcome, Alan.
Thank you. Pleasure.
All right. And next up, we have Alex. Hey there, Alex.
Hi, Laura. How are you?
Good morning to you. And he's based over in Ohio. And he's been with Bio-Rad for 6 years. So another great mind to join us on the panel. And he's been focused on supporting people with flow cytometry. So welcome, Alex.
Thank you.
All right. And last but not least, joining us from the city by the bay, San Francisco, we have Brandon. Hey, Brandon.
Good morning.
Good morning. And Brandon's been working at Bio-Rad and supporting the cell sorting portfolio for the last 5 years. So welcome there, Brandon.
Thank you.
All right. So I think we have everybody's audio and video is coming through nicely. And so before I kick us off with a couple of warm-up questions, I just want to let the audience know that we love to get your questions. So please do type those into the Q&A and we will get them across to our panelists. And don't worry, if you're listening on demand, we can still get you those answers. We'll just reach out to you through the email address that you use to register for the event. Okay, panelists, are you ready? Okay, let's get going. So I think this is a really good question to get us started. All right. What is the best method for cell dissociation? Is it cell scraping or trypsin? Who would like to kick us off? All right, Alan.
So I would actually temper that away from scraping and trypsin. Scraping is very damaging to the cells and can possibly kill the cells, so you get less end product from your sort. Trypsin can be damaging to epitopes on the cell surface, so I'd recommend something more like a collagenase base like triple LE or acutase that will disassociate without damaging the cells. Also, you can use EDTA, 2 to 5 millimoles as starting, and this will help disrupt the gap-to-gap junctions of the cells and also stop them reclumping and clogging in machine.
Got it. So no scraping and no trypsin. Beautiful.
Preferably, yeah.
Brilliant. All right. So anyone have anything to add on that, or did Alan get it? All right, we all agree with Alan. I like that. I like that unanimous decision. So next up, we have another question that kind of follows on from that one. So what's the best way to overcome or prevent clogging if someone's getting lots of clogs with some of their samples? And I don't believe that's a reference to Holland footwear, but who would like this one? Brandon.
Wow, nice reference.
Bless.
Yeah, so clogs. Yeah, a cell sorter's worst nightmare. I would say probably the first thing you want to do is you want to make sure you're using a cell strainer. So yes, that's a real thing, and that'll help you remove the clumps out of your sample before you actually load it onto the sorter. And you want to make sure you do that just prior to loading onto the sample, loading the sample into the sorter as well. You know, it might be that you're working with some really sticky cells though. And if you're running like a long sort, they may tend to start clumping actually within the sample holder. In that case, you know, actually, as Alan had mentioned, you want to make sure you add EDTA, something between 1 to 5 millimolar concentration within your sort buffer, but you don't want to add too much because EDTA actually can be bad for cells at higher concentrations. And actually, an EDTA might also could potentially affect some of the cell-based assays you might be working with. So if that's the case, there are alternatives like sodium citrate or even using like PBS that's free of calcium or magnesium. The other thing too is if you have a really terrible sample, like it's got a lot of dead cells in it, or it's got like a lot of free floating DNA, you might want to add DNase too as well. Something like, I don't know, 10 units per mil is pretty good starting concentrations to kind of help chew and grade up that DNA, because that will make the sample sticky as well. I think also you want to pay attention to the concentration of your sample. So if it's too concentrated, that could actually gunk up the uptake. So you probably want to shoot for something around the range of like 10 to 15 million cells per mil in your tube if you're using like a 100 micron nozzle. And then, and that's the other thing too. Make sure you're choosing the right nozzle size. Some people may not know that you want to aim for like 5 times the size of the cell diameter you're actually sorting. So too small of a nozzle size can result in clogs as well. And then finally, take care of your instrument. I try to tell this to everyone who's starting their journey on cell sorting. Good cleaning protocols, make that habitual. So at the end of the day, make sure you're cleaning your instrument and make sure you're using the right cleaners. Some are better suited for analyzers like CultureCleanse, and some are just better for cleaning out the debris and whatnot in the cell sorter, like Contrad. So maybe you want to think about using Contrad.
Whoa, that was a lot. Thanks, Brandon. That's brilliant. Did he miss anything, panelists? Oh, I think maybe one thing there, Chris.
Just the whole. No, it's just one thing. Is Brandon's right in all those aspects, but one thing to remember is if you are going to sort, there's a temptation to put serum back into your sorting sample, and if you do that, you're going to add a lot of protein, which is good, but you're also going to add a lot of free ions, which can actually help make the cells stick back together again. So if you are going to use some kind of protein in that sorting buffer, it's better to use BSA or something like that. But because there's a lot of unknown stuff in fetal bovine serum, so just bear it in mind.
Brilliant. All right, so now let's get on to some audience questions after we've warmed up those brain cells. All right, so this person is writing that their sorts aren't as pure as they need them, and they're only getting 60% of their target cell. What can they do to improve? Alex.
Hey, so one of the things that I've seen a lot is people would take their post sort and run it directly without washing the sample first. So what you would see is a lot of the debris. So what you might find is once you do wash the sample, just take a little aliquot of the sorted, you would find that your purity is indeed higher. If that still doesn't work, then I would start looking at your drop delay just to verify that is correct.
Brilliant. Anyone want to add to that one? Nope. Okay, Alex got that. Nice one. Oh, no. Alan.
I would also pay attention to your gates and how you've gated your samples, because if you've gated them incorrectly, you're going to pull some incorrect cells into your sample as well. So make sure you've gated your sample in the correct fashion so you are selecting the cells you want.
Brilliant. Oh, Chris.
Yeah, one other thing to pay attention to is the compensation as well. Making sure that you've got a good separation between your positive and negative cells so that you're not having any overlap. So getting good single stain controls and FMO controls if you've got a bigger panel, just to make sure you can actually really pull out that population and you're not pulling anything which has some spectral overlap with it would be quite an important thing. Yeah, I think a viability dye would also be pretty handy in this situation. If you're pulling out dead cells, they can attract some non-required items into your sort. So it's good to have good, clean, healthy single cells when you're sorting.
Brilliant. All right, so another one from the audience here. And I think that's a good follow-on to the discussion you're just having. So any tips for preventing contamination of sorted cells? Alex.
So you might probably want to make sure that your instrument is, as Brandon mentioned, as clean as possible. So I recommend, running maybe a tube of bleach and then cleanse in water before you sort, and then also after you sort, just so you leave it, in good shape for the next person. So, yep.
Brilliant.
I would also add to make sure your tubes are capped quickly, wipe everything down with alcohol inside the sort chamber. Make sure you use good sterile techniques when pulling out the tubes from the instrument and the area within.
Got it. Brandon.
Yeah, I just wanted to add that if you're not confident, if you don't think that you're actually getting all of the potential contaminants removed from the system, pretty easy way to tell if you are getting contaminants, just to collect some of the stream in some LB broth or PYE to see if anything grows in that as well, and let that sit overnight. And that should tell you whether or not you actually did a really good job of decontaminating your system.
Mm-hmm. Yeah.
And you can also sort into antibiotics, penicillin, streptomycin, so you can start inhibiting it early on if you're worried about contamination as well. And a lot of cells and cell growth media have that in them. So you can always add that into the collection media.
Brilliant advice. All right, let's get us moving on. So this scientist is asking, should you add visibility dye again to the sample you are rerunning to test purity? Chris.
I think, the short answer is you can. And there's a whole bunch of dyes that you can add that you don't need to wash off. So some of the 7AADs or PI or something, it's a real-time assessment. The ready drop ones for WeCell, for instance, you just add a couple of drops into your sample, and as you'd run that through the sorter, you'd get a live, a real-time image of what cells are alive and what are dead. Now, that won't go, well, you'll get some transfer into your collected sample, but you really want to sort of add another couple of drops to your collected sample when you rerun it back through the sorter. Now, saying that, if you're only running a very small amount, you're going to have issues with how many, one drop per, however many cells it is. But if you're not interested in how many live cells are in there, then you don't need to add a viability dye if you're just, looking at something else that's downstream or whatever. So that's not necessarily about purity, but it would give you an indication of how good your sort is, or how, if you've been sorting for several hours, you may end up with a whole bunch of dead cells. So it's good to get those taken out.
Got it. Gentlemen, anything else? Nope. All right, let's move us along. So, all right. This person, a general challenge with sorting that I experience, the yield beyond the purity that has been mentioned. How can one increase sorted cell yield beyond increasing the initial starting material? Take a second to read that one. It's quite long. Brandon.
Yeah, without knowing, what kind of conditions the cells are in before going into the sorter, I think one of the first things that comes to mind is you really want to pay attention to how accurate your drop delay calculation might be. The drop delay will actually, so a bad drop delay will actually affect the yield more than purity. So you may not actually even realize you're losing a lot of cells if that drop delay calculation is not right. Preferably you want to choose a system that is able to actually get a really accurate drop delay calculation, like the S30 cell sorter. Just wanted to drop that in there. The other thing too is you just want to make sure that you have a really healthy sample before you're loading it onto the instrument. It could be that a lot of your cells are dying. There's something you're doing treating the sample before you actually put it on the instrument that's causing a lot of cell death and whatnot. So just remember that it's always, it's a mantra we like to say in the flow cytometry world, which is garbage in, garbage out.
Alan.
I would also look at what you're sorting into. So a lot of people may just think you sort into your 10% media, but as you're sorting, you're diluting your cells and into that media. So you're losing less nutrients. So I'd start off in a higher rich nutrient media to sort into. In addition, I would do something that's called pre-coating the cells. So as the cells go in, you've got static and protein binding sites on the tube. So sometimes that will actually bind up some of the cells going in, reducing your yield. And plus, in addition, you're going to get attrition of your cells going into a sort anyway. So your yield's going to be slightly lower than what you've collected just because it's quite a harsh thing you're doing to a cell, sending it down the stream and basically belly flopping it onto a tube. So you're going to get some yield loss, but like Brandon says, think about what you're collecting from and what you're collecting into to help it make them happy.
Oh, let's do Brandon, then Chris.
The other thing I forgot to mention is you might be sorting too fast, like the event rate might be too high. So it could help to actually decrease the speed a bit. If you're trying to run at the max event rate of a sorter, you're going to decrease the efficiency of the actual sort. So if you want to potentially lower the sorting speed or maybe even reduce the concentration of the samples, that could also be an issue. You might be creating a bunch of coincidence events just by having it too concentrated. And so a sorter will abort a cell if it can't actually determine whether or not it's a single cell. So a couple of other things to think about.
Mm-hmm. Chris?
Yeah, I was just following on from that with the coincidence events with the single cells. Really make sure you've got good sample prep in the first place. So if you do have even two cells stuck together, that you're going to abort that. So it's really important to make sure. And you can check under a microscope when you've done your cell straining. Take a sterile sample, put it on a slide, stick it under a microscope, make sure you've got some nice, good single cells, good dispersion, the concentration looks good, etc. And that's really the best sort of look-see to see what's going into your cytometer.
No, that's great advice. Alex?
So along the lines of what Brandon mentioned about sorting speed, I've seen a lot of times, especially in core labs, where people just don't book enough time. So they're trying to sort in 30 minutes, what really they should have booked 2 hours for. So, be mindful of, what time you really need for your sort.
Yeah, don't rush the science, don't rush the process. Totally. All right, let's get us with another question here from the audience. So any suggestions for what quality water to use when making 1 times sheath buffer from 10 times buffer? Alan.
Basically high quality, good, the 18 megaohm water, good filtered water is very good. We actually sell salt grade water, which can be used to create your buffers from. But any good molecular grade 18 megohm water, please make sure your system's checked, because if it hasn't been checked in a while, you may not be getting as pure water as you think you are. So good molecular grade water, good 18 megohm water from your water purification system should be good enough to make your buffers. And then before use, you might want to think about filtering them, a 0.2 micron filter, just to make sure you have no particles going forward into the system.
All right, great advice there. Next up, oh, here we go. Every time this person sorts into plates, their cells die. Why? This sounds like it could be a long answer, but let's see. Brandon.
Yeah, that's another tricky one. I guess the first thing I would ask is, so just like you're sorting into tubes, are you sorting into capture? Is there capture buffer in the plates? You know, you want to at least probably have something like 200 microliters of capture buffer in your plates, in each well. Are you sure the cells are actually hitting the center of the well and not, you know, maybe hitting on the side of the plate? So, or just outside of the well. So you might want to just double check that. The other thing I could think of maybe is that your cells just don't like to grow individually. You know, there are some cell types that they need to grow with other cells to remain viable. So for example, like stem cells. And in that case, you may actually have to just sort on something like a matrigel and pick colonies if you're, you know, doing something like CRISPR gene editing or any kind of other clonal expansion.
Yep, got to keep those cells happy. Okay, let's get us to, here we go. What temperature should I sort my cells at? Oh, this is a two-parter, and is it okay to sort into 10% FBS? Alan.
So it would depend on the cell line and what downstream applications you need. If it's maybe if you want to cell culture it straight away, you need to increase the temperature a little bit. If you are putting into a lysis buffer, you may not need to change the temperature. I would definitely check the literature to see what's been done in the past, particularly if it's been successful, then you can follow their protocols and what they've done successfully. So that should help you out. In regards to 10% FBS, like I said before, you can sort into 10% FBS, but because you're diluting it during the sort, I would increase that to 50 to 100% FBS. Give them a nice, good nutrient-rich media to sort into. So starting off with a higher amount and knowing how much you're going to dilute it in will help keep those cells, that viability, that yield up, and the post-sort.
Brilliant. Oh, Chris, want to add? There you go.
Yeah, just add a little bit. If you think about it, a lot of mammalian cells are all designed to sort of be nicely incubating around 37 degrees. So it's great to sort of if you're looking at things like receptor turnover or enzyme activity and you want to slow that down, you don't want to lose any signal. And as Alan was saying, you know, check the literature on that. But things like PBMCs, for instance, they're really happy at body temperature, and they might actually not like being sorted at 4 degrees. So unless you're really interested in something specific further downstream that, you know, when you're sorting, you're losing that molecular activity or enzyme or some kind of cell receptor, you might want to think that 37 is actually not a particularly bad temperature to sort at, and it gets a bad rap, but PBMCs, for instance, are pretty happy at 37. So, yeah.
Got it. So consider the cell type and downstream what you want to do with your sample. Brilliant. All right. Let's keep our brain cells going. So this person, okay, so sorting speed is a challenge. How can one strike the balance between some cell sorting that takes very long? Ooh. Lots of coffee breaks, Netflix. Other advice from the actual experts? Brandon.
Trying to think, maybe I'm camouflaged here. One of the things you might want to try actually is just doing like a debulking prep step first. So, run it over like a max column, and you potentially could remove a good amount of cells, and that could help you reduce the time, the actual sorting time.
Any other advice? No. Ooh. The panelists are stunned into silence. All right, let's see if we can wake everybody up with this one. This person keeps getting goodness. This person keeps getting contamination in their sorts. How can they better prevent this? We might have touched upon these already, but Chris.
I think we did that one already. I think you have a circle back, isn't that? We're talking about, if we haven't, I'll bring it back up again. Make sure we've got a good sterile technique when you prep your sample, and make sure your instrument isn't contaminated in the first place. So checking your stream, and make sure you're cleaning your sample, cleaning your machine, and running a decon if you actually have access to that program. If you have seriously bad contamination, you have to run a decon cycle, running bleach through your system for a period of time, and Contrad and all these other things that we've talked about to dissolve proteins and get rid of bugs. But yeah, I have a feeling we've had a very similar question.
Similar question. Got it. All right. Brandon.
I just wanted to quickly add that, if you're having trouble with contamination from, say, maybe users sorting bacteria, or you're using a system that's shared between mammalian and bacterial sorts, you might actually want to think about buying your own microbial designated sorter. So just throwing that out there. I realize it's probably not the solution for everyone, but it is definitely a route you can take.
Mm-hmm. So how to keep your other lab members happy, right? Just get your own and stop messing theirs up. Got it. Yeah.
Exactly.
That's good advice. Alan.
Also, some of the contamination may come from your own buffers that you're using. I had it once where somebody's water was clean, and the person who took another aliquot from the same bowl, their water was very contaminated with bacteria. So check your buffers that you're using, make sure they're clean and sterile before adding it to your cells, because that can be a source of contamination, is all your buffers pre-hand going in could be not quite as clean as you think they are.
Mm-hmm. Yeah, do the old pick up the bottle, give it a swirl, right? Make sure there's nothing growing in there. Yes. Oh, Brandon.
There was just one more thing I wanted to add, I think, and I alluded it to previously when we first started this, is you want to make sure you're choosing the right cleaning agent. So, my colleagues might be able to correct me on this if I'm wrong, but I think, for example, let's say that you're working with, viral particles. So, for example, you've got like some kind of phage transfection or transduction. You might want to use ethanol as a cleaner in that case rather than Contrad. So just a thought. So maybe they have some additional insight around that. But again, the right cleaner for the right cell type or the right kind of sort.
Got it, Alan.
So yeah, for that, like bleach and ethanol tends to clean some more. There are some antiviral cleaners you can run out there. So those tend to be killing. Like you saw, you can have your waste going directly into bleach, so it kills things at the back end. But making sure you're using cleaning agents that kill for the contamination. Things like Contrad is more of a detergent base, so they tend to clean things. So like when you wash your hands, you go through a detergent. So when you clean your sample line, you want to try a little bit of a detergent too. But Brandon's right, you want to go for those killing agents if you've definitely got contamination. But always flush the line with copious amounts, like 3 times the amount of water afterwards, just to get those remnants out, because you don't want them touching your cells. It's going to be a little bit detrimental. So a lot of flushing of water afterwards would be great.
Mm-hmm. Yeah. Yeah, because some of these cells, you might not be able to go down the hallway and collect some more that easily. So good system prep is important. Chris?
I would also say don't cough, sneeze, lick your samples. They don't hug them. They don't like it. They like to stay clean. So
I love
keep
it.
that tube covered. Wear your mask at all times.
Love it, love it, love it. I can see some posters going up on the wall in Chris's lab for this one. Oh, Alex.
So just to add what Chris was talking about, I have seen labs, they're using the same lab coats that they were using for, animal work, and they're passing it right over their tubes. And, they're asking, why is there contamination? I said, well, maybe you should try finding a separate lab coat, So.
Got it. So don't scrimp on the supplies even. Yeah, it's all important. Got it. All right, folks, let's move us on to another one. That was a lot of good discussion there. Ooh, how fast can I sort, and does speed really matter? I mean, if you ask Keanu Reeves and Sandra Bullock, I'm sure they'll say yes. But let's see what the panel. I think Chris, go ahead.
Yeah, it's a real balance. Does speed matter? It depends. If it's 7 o'clock at night and you really want to get home, I'm sure speed matters a lot, especially if it's Friday, there's a cold beer in the fridge, But saying that, if you are sorting really fast, there's a chance you're going to A, damage your cells because they're being subjected to higher hydrodynamic forces. B, you're going to get a lot of coincidence events, which I think we touched on previously. And when you're doing that, actually, when you're sorting faster and you're actually creating a lot of turbulence, you're getting these coincidence events in your drops, you actually might be sorting slower at the end of the day, because if you'd sort a little bit slower, you'd actually have more events coming through into your sort and not being sent off to the translation, waste paper basket, trash, junk. That's a real an issue. And also, if you're sorting fast, damaging your cells, the cells that may even get sorted are not going to be very happy, and they may end up dying later on down. So if you've got a downstream process that requires a lot of live cells, it's a really good idea to slow that sort down, take your time. Obviously, if you're sorting something like bone marrow stem cells, they're a very, rare population. The sort's going to take a heck of a long time if you're trying to do it really slowly. I'm talking 3 or 4 hours. Cells don't like sitting in buffer for 3 or 4 hours. So it really is going to be almost a trial and error at that point. And you're going to have to find a balance between cells sitting in a non-ideal sorting buffer versus getting them back in the incubator and getting them happy and growing, or whatever downstream process you're looking at. So the general recommendation is about 1 cell per 4 drops. So you don't want cells in your drops in your drop today. Yeah. So you don't want cells in the first 2 drops. You want a cell in the middle, followed by no cells in your following 2 drops. That way, you have a nice sort of spread of your cells, and you don't get these coincidence events. And that it all really comes down to sort of speed and concentration of your sample, and finding a balance in that.
It's all about the balance, right? Alan.
It also depends on what you need. We have customers that really don't care about purity, so they can actually increase their speed because they don't mind those coincidence events coming in. But I agree with Brandon, once you start wanting more pure and better efficiency, you need to go to that, look at the frequency of your machine, go to 1 drop every 4 drops, and then you should get increased efficiency. So increase your yield and keep your purity at a high level.
Fabulous. All right, let's get another one from the audience. Okay, this should be an easy one. What % Contrad do you use for cleaning? Brandon.
I think somewhere between 2 to 5% is probably going to be okay for you. That's sort of a recommendation. I've heard of some people using a bit higher, but I would generally recommend staying within that range.
All right, short and sweet. Next we have, I guess it's a related one here. So what's the best way to clean a nozzle if they have a clog? Alan?
That's going to depend on the instrument and the nozzle itself. For the S3E, the software has some nice things built in, like low and high pressure cleans that will force cleaner through the nozzle to help flush out some of the clogs that you have in there, minor or major clogs. In addition, you can actually remove the tip, and for us, you can use a syringe to pass water through. For most of the tips out there, you can put them in a cleaning solution and put them in like a jewelry sonicator, which will then vibrate and dislodge some of the clogs from the nozzle. But that's generally the main way, is to take it out, flush it, sonicate it to try and flush those clogs out of the nozzle tip.
Perfect. All right, I see no other hands raised, so I'm going to get us on to the next one here. We're getting through them fast and furious. How robust are fresh blood cells during sorting? So for multiple cell types, is it better to do this as ones with multiple staining, or okay, so for sorting multiple cells, is it better to do it like that, or with multiple staining, or with multiple steps? That's another long one. Chris?
Yeah, I think that depends on your sorter. If you can do all of your sort in one go, so you have access to an 8-12 color sorter, you go for it. You know, add the antibody in and get sorting less times is better than doing a stepwise sort, because each time you pass your cells through your sorter, you're going to have subject those cells to those stresses and potentially damage. Saying that, if you have a 2-color sorter and you need more than that to get your cells out, there are other ways that you can sort of get those cells down. Brandon mentioned it earlier with the max sort, subjecting them to some magnetic beads, try and reduce the population a little bit to get closer to what you actually want. But at the end of the day, it comes down to what kind of sorter that you've got access to. So I think that's important to remember. But yeah, if you can get those cells that you need within one sort, that would be much better than running through the sorter multiple times. Saying that, B cells, T cells, it could probably take a couple of sorts, but you are going to lose some viability down the line.
Alan?
So fresh blood cells, like I said, are fine, but you may also want to think about isolating the PBMCs, the white blood cells, beforehand to help you focus on the cells you need, or using like a red cell lysis buffer to get rid of the red cells. So you're more focused on the cells that you want to stain and sort rather than excess of red blood cells that will overwhelm the system over the PBMC. So think about trying to isolate the cells from it first with like a isolation PBMCs or something like that, red cell lysis buffer. That'll help you focus on these cells.
Perfect. Okay. All right, so this person is trying to sort primary mouse hepatocytes using GFP, but they're not getting enough and they keep clogging the instrument. Oof. Can you recommend anything? Chris?
I'll take that one. I have a lot of experience with primary mouse hepatocytes from a former life. Yeah, they're big and they're sticky and they're, very, very troublesome. However, it really comes down to that initial sample prep. I think we're touching on this a lot during this coffee chat. Brandon mentioned it, rubbish in, rubbish out, or whatever he said in American. Trash in, trash out.
Yeah, something like that. Translation.
Translation. So yeah, it really comes down making sure that you have a good single cell digestion. So subjecting to breaking those cells up with some good collagenases and other enzymes, checking it under a microscope. You don't want to over digest your sample because you are going to start to damage the cells themselves. And that is only going to come with experience and time and trying to figure out, have you getting the right conditions for experiment. look in the literature and see what other people have been doing before you. And that's really going to be your best resource. And I think once you figure that out, make sure that your sample is good under the microscope before you put it into the cell sorter. You might still end up with some clogs, but that's the risk you're going to take. And strain the sample using the cell strainers and make sure the concentration is good, not overly concentrating primary cells.
All right. Oh, Brandon, something to add. Okay. Good. You might have to translate it for Chris, though, I think.
Yeah, rub-ish. That's like almost rubber, right? It's just rub-ish.
Rub-ish, yeah.
Dad joke. Sorry.
Dad joke.
As Chris had mentioned, one other thing to think about is make sure you're using a large enough nozzle. So I think Chris said if it's a large sample, if it's a large cell size or it's a large cell diameter, looking to use something like 100 micron nozzle is probably is more suitable for that kind of a sort.
Got it. All right. Oh, Chris.
Just one more thing is, yeah, there's multiple washes as well. If a primary cell sample is going to be more dirty in lots of aspects than a nice cell culture sample. You're going to have a lot of serum in there, a lot of albumin, lots of other things for complement system, etc., coming through. So once you've isolated those cells, wash, get rid of all those binding agents and extracellular matrix. Make sure it's all gone before you run your cells through the system.
All right. Thank you, gentlemen. So next up, this is an interesting one as well. So is there anything I need to take into consideration when sorting bacteria that is different from mammalian cells? Brandon would say get your own cell sorter, but Alan?
So there are some things to pay attention to because bacteria tend to be a little smaller than cells, so they may not get too far out of the debris or the noise. So good sample prep, not having it in necessarily LB broth when you're going to sort because that actually adds more noise. Need to change the threshold of the instrument to allow the signal from those bacteria to come through. Also, what sometimes helps is if you have a fluorescent marker on the bacteria, you can do what's called backgating, where you trigger or you just look at what's glowing, say, for cyber green, looking at DNA, and then backgate into your forward side scatter plot to try and locate where your bacteria is in those other plots using a fluorescent marker. So it's easy to find them using a fluorescent marker. In addition, it goes back to both what Brandon and Chris have been saying about concentration. If you have too much, if you lift it up and it's very cloudy and you can't see through it, you've probably got too much in there. So you'll want to dilute bacteria generally a lot more than you do cells because they're smaller, they'll have a greater concentration. So if it's generally a little too cloudy, you'll probably want to decrease concentration. And always decod after use, always clean the machine after use. Leave the machine how you want to collect it in a clean, uncontaminated manner.
Yeah, there's nothing worse than spending weeks and weeks, growing things up and then going to use an instrument and it's all, yeah. Okay, let's not talk about my nightmares in the lab. Anything to add, anybody on that one? Nope. All right. Here we go. What is a good live-dead ratio for bacterial sorts? Chris.
Yeah, I think that's talking about the looking at a viability dye, basically, is how I interpret that. I think bacteria a little different to mammalian cells. PI would work. It would work in bacterial cells as it does in mammalian culture. However, it isn't just about having an intact membrane with bacteria. So just because you add a PI in and it's not being taken up by being able to get through a permeabilized membrane of bacteria doesn't necessarily mean that that bacteria will grow further down the line. So it's important to look at other factors. It's viability and vitality as well. So you need to look at things like dye exclusion, active enzyme dye exclusion. So back in the day, you could add ethylium bromide and make sure that the bacteria are not taking that up. So yeah, you need to make sure the bacteria do have non-permeabilized membranes, but actually some bacteria that have permeabilized membranes will actually grow. But also that their enzymes, they're on the surface, the cells themselves are actually working and functioning normally. So
a bit of a tongue twister there.
Yeah. Too much coffee.
Too much coffee, right? Anything to add, anyone else? Nope. All right, let's get going. We managed to squeeze a few more in here before our time is up. All right, this person is losing their target receptor over time while their sample is sitting in the sorter. What can they do to stop this? For example, their positive population is decreasing. Chris.
So it helps if I take myself off mute, and that way my answer probably makes more sense.
Yep, definitely.
Sorting at 4 degrees would be a good one, be the first thing I would say. Receptor turnover is going to happen. Going back on what I was saying earlier about sorting at 37 degrees, I'm now changing my mind. In this particular circumstance, trying to sort cold, so you're going to slow those metabolic processes down, and making sure that you've got a nice buffer. Sorting in PBS is all well and good if your cells can take it, but if you need lots of protein or the cells aren't happy in that buffer, then changing it and figuring out what would be a better, more suitable reagent buffer for them to sit in and make them more happy. Sure, people have got other things to add to that.
Brandon.
Yeah, just a couple comments. One, just to reinforce what Chris says, cold stops capping and shedding, so something to think about. The other thing too is, as I alluded to earlier, is it depends on, there could be some component of your buffer that's actually causing this. So I think in some cases, EDTA is known to downregulate some receptors. So you might want to think about, switching out some of the contents of the buffer, and just kind of targeting and removing the variables to see if maybe that's what's actually having an effect on your sample.
Yep, play around with the parameters and see what works best. Good advice. Good advice. Alex.
So the other thing you might want to check, I haven't seen it much. So most sorters that I have seen are in dark rooms where I have seen one that they had it right in front of a window. So if sunlight is hitting your sample, you just might be losing the fluorescence because the sun's just basically bleaching your sample.
So kind of like a vampire, keep everything in the dark. Got it. It's the vampire effect. I'm sure that's not a scientific term. Please don't let it get out beyond this coffee chat. Here we go. All right, so this person, their sort is going well, but when they do a post-sort purity run, they're getting low numbers for yield compared to sample input. Any suggestions of why this might be happening? Alan.
So you will get some, like we talked about earlier, some loss of cells in the sort. I generally aim, if I want 100,000 cells, I'll try and sort 150,000 just to make sure I have plenty of cells. Coat your tube so there's no loss of cells binding to the side of the tubes. Make sure, like Brandon says, you have liquid in the tube to sort into and make that high nutrient rich, either with FBS or other nutrients. Really due for your stem cells and things may have other needs. Check your gates and other things. These all impact your purity. Make sure you selected purity in your sort logic or what the logic of that sorter. If you sorted for enrich, your purity is definitely going to go down. So make sure all your checks and balances in the software of what you want to sort, you've taken care of, and then take care of your sample as well.
All right, gentlemen, anything else to add on that one? No? All right. Well, we've got a couple more to run through here. Here we go. Is it okay to run samples reconstituted in media containing FBS, MPP media, as compared to only 1 times DPBS? Oh, that was hard to read on my screen. That's like an eye chart. Alan.
So media is good to run in, but I would also recommend phenol red-free media. The phenol red is going to actually light up and create a lot of background noise from the media itself off the lasers. So if you have to run a media containing FBS, I would recommend having the phenol red-free media. But I would also add to the PBS, the Dobecco's PBS, so either 2 to 5% FBS or like 0.2% BSA to add protein into the reconstituted cells just to help keep them stable as well. So media and FBS is good. No phenol red, but also PBS with a little bit of protein is good as well.
Super. Anyone want to add to that? Oh, Alan, you got the whole thing there, Alan. Nice one. All right. Stunned into silence. Here we go. Ooh, so this person is asking their post-sort viability is low and they really need it to be higher for their downstream applications. I think my cells might not like to be sorted. What can I do? Oh, Brandon.
That's understandable. Sorting, and depending on the sorter type you're using, can be kind of harsh on cells. So, but there's definitely a couple things you can do to make sure that you retain viability throughout the sort process. So, obviously, we talk a lot about sample prep. One thing I would definitely make sure, or would highly recommend, is to include a viability dye when you're building your panel. You have a couple of options. I think, we prefer 7-AAD because it's a bit more of a late stage apoptosis marker. So you want to make sure you're catching those cells that are already well on their way to dying. And so they're not dying in the tube after you've sorted them. Make sure that you've got really good sample going into the sorter. Again, rubbish in, rubbish out. Right temperature, right? You might want to think about how you're prepping the sample before sorting. So Alan, as he mentioned, scraping is really, really bad. You're gonna basically try to run a pretty junked up sample if you're having to scrape adherent cells or whatnot. And it could just be that you're actually working with a sample that doesn't go very well in the single cell suspension. So if you're doing something like single cell analysis, you may want to think about just sorting nuclei instead. Make sure the right nozzle size, the right collection buffer. Really think about how long the sort's going to take. Alex actually mentioned this as well. Depending on how rare the population is, or like if it's a low frequency population, you could end up sorting for 4 hours straight, depending on how many cells you need. So the length of sort can really impact the viability as well. So you might want to potentially, again, so debulk your sample first, using magnetic beads before you run them on the sorter. And then finally, I'll probably catch some flack for this, but I wholeheartedly believe that jet and air systems are much more gentle than cuvette-based sorters. So you may want to favor using a jet and air system over a cuvette-based system. Cuvette-based systems are really great. They're better when it comes to the resolution. But I wholeheartedly think jet and air systems are much better, much more gentle on cells during a high-speed sort.
Mm-hmm. Not
and guess what? The S3E cell sorter is jet and air system.
Oh, blimey.
So, think about that.
Product placement. There you go.
I just wanted to add, hey, we're having a promotion on the S3E right now as well here in North America. So make sure to follow some of those links that we have available for you and direct yourself right to the S3E cell sorter page.
Shameless.
Shameless.
Absolutely shameless. But hey, it's all good. All right, here we go. I kept this one for last because it's pretty funny. It's late. I want to go home. How important is it to follow the shutdown and startup procedures? Alex.
So again, you want to be mindful of the people that are using it after you. So it's always good to be courteous, and you really never know what happens if you encounter a global pandemic and you don't know when you're going back to the lab. So it's, a clean sorter is a happy sorter, is what I'll say.
Oh, that is hilarious. I just went camping, and I agree with that. Leave the camping toilet as you wish to find it, right? Because you never know when a global pandemic will be happening and everybody's going camping. Anybody want to add to that? Alan, and then Chris.
So yeah, leave it as you want to find it, because that little extra time putting in for a little bit of cleaning at the end of the day or first thing, you may not then have to go and clean your nozzle, take it off, spend the time talking to tech support, realigning your instrument. That extra little bit of time up front can actually save you a lot of time on the back end when trying to restart your instrument or for the next person starting up their experiments.
Yeah, you don't want to be that person in the lab, do you? The one that everyone's like, oh, God, so-and-so used it. Now we're screwed. Okay, Chris.
Yeah, just I think we've mentioned it before, but when you are following the correct shutdown procedures and you're loading in your Contrad or your bleach, it's tempting to go, I've just run my bleach there, I'll just leave it. No one will know. Some of these agents can be pretty harsh to the internal workings of a cell sorter, so wash with water a couple of times, make sure you rinse it out well, especially if you've been running an infectious substance and you've run your antiviral through there, or your ethanol and bleach. You don't want those detergents to form crystals inside the machine or cause other problems. So wash it out with water and shut it down, and everyone will thank you later.
Beautiful. And I know I said that I was saving that one till the end because that was the funniest one. One more has snuck in, so hopefully everyone has a couple of brain cells left to rub together. Here we go, and it's a long one. Here we go. So when performing single-cell sorting on highest purity mode with filtered iPSc cells without a viability dye due to toxicity, one still observes several cells in a couple of wells in a 96-well plate. How could this be improved or avoided? And that's a long one, so. Alan.
So just statistically, you may get 2 in a well, but if you want to really home in on that, use the highest purity mode. Decrease your concentration and speed. Allow for at least that 1 cell per every 4 drops, if not more, to allow yourself that time in the sorter to give yourself that separation of cells in that. And then it goes back to that preparation. Single cells in will give you single cells out. In addition, make sure you're gating out those doublets. If you don't have that doublet gate, there's a highly likely that you're going to get doublets through there. So make sure you have your single cells, gate out your doublets, and go a little slower and maybe a little lower in concentration would help. In addition, you can optimize your drop delays to make sure that's going at the right place. Redo your drop delay, make sure that's been optimized for your cells and your instrument at that time.
Okay, Chris.
Yeah, just one quick comment on the doublet gates. It's important, I think, and I've got into the habit of doing a forward side scatter, forward doublet gate, forward scatter doublet gate. Sorry, I'll get it right in a second, as well as a side scatter doublet gate. It depends on the instrument, but side scatter is sometimes a little bit more accurate in terms of doublet discrimination. I like to use both. Why not? Doesn't cost anything. And you'll find that you'll probably get some more single cells come out if you use both of those gates. So yeah, that was just it.
Thank you. Oh, Brandon.
I was just going to say, so for the individual who asked that question, I was actually reading a paper recently about optimizing plate sorting for single cell analysis applications. So if you want to email me at Brandon_Williams@biorad.com, I'd be happy to send that paper your way, and that could potentially offer some tips for you.
Brilliant. We'll get that connection made post-event. All right. Okay, that's it. No more questions. You can all breathe a big sigh of relief. You did really well. Shameless plugging. We'll look over that. We are
I am the
out.
product manager.
That is true, right? You are the product manager. It's a little baby. So without keeping anyone on the line too much longer, I just wanted to give a shameless plug myself to some of the other coffee chats that we have in the series. We have quite a few already recorded, so you can listen to those on demand, qPCR and infectious disease. There's another one that we did around cell analysis as well prior to today's event. And we have a couple more upcoming too, for example, on the ddPCR workflow. So please listen to those, sign up, register. And if you have any additional questions, please feel free to reach out to us. We love to get your questions. And with that, thank you everybody on the panel. And thank you to the audience for questions. And I'm going to sign us off here. Take care, everyone, and stay safe.